Hello again. So we've been discussing the ACM components, generally the ACM and its function and its role, and then we started discussing its components. The fibrous component we discussed last week, and we discussed um, the major fibrous component of ACM, the collagens. Today we'll be talking about the squashy gel part of the ACM, the sugars. So the carbohydrate component is usually found in the form of glycosaminoglycans. I told you before I cannot quite say this way too many times, so I'm going to be saying GAGs from now on, or proteinoglycans. So GAGs are similar to polysaccharides, so that means that they're repeating unbranched chains of processed disaccharides. Disaccharides means that it's made of two sugars, okay? Di at the beginning of the word disaccharides means two, and saccharides means sugar. So um, each component of a polysaccharide is made of a processed disaccharide. They are highly hydrophilic, so that means that they like to actually absorb quite a lot of water, and they're negatively charged. So GAGs are very large complexes, and they can be associated with a small amount of protein, and when that happens they form proteoglycans, which typically consist of more than 95% of the sugar, of the carbohydrate. GAGs have the special ability to actually bind large amounts of water. So this is because they are so highly hydrophilic, and thereby producing that gel-like matrix that forms the basis of the body's ground substance. The viscous lubricating properties of mucus secretions also result from the presence of GAGs, and this actually led to the original naming of this carbohydrates as mucopolysaccharides. So here I'm showing you with this red dotted line the disaccharides which make up this GAG. So basically, some of the properties of and roles of GAGs, they are able to occupy large volumes relative to their mass, and a good example is hyaluronan or hyaluronic acid, as it's called. And you can see here that compared to its molecular weight, it occupies quite a large. It's able to occupy a large volume as opposed to other molecules found in the ECM. They are found generally in connective tissues and in the extracellular matrix of the connective tissues, and also they are mostly found on the surface of many cells. So when they are actually made by the cell, they stay close by at the surface of the cell. They form gels and are highly resistant to compression. And a good example of that is the fact that we see a lot of it in our joints. They have this slimy mucus-like consistency, just to give you an idea of how they actually look, what type of consistency they have. And they are also space fillers in development, into which cells can migrate around in the embryo and locate themselves in areas where they can start forming the organs. They provide high viscosity and elasticity, and that actually it's very important for the roles and for the tissues that we find them in. When the GAGs are associated with a small amount of protein, they form proteoglycans. So proteoglycans, which means that there is a protein in a GAG, and their constituents, both proteoglycans and their GAGs play diverse roles in ECM. So they're usually secreted, and again, they're found in the cell surface, and they can actually stay in the cell surface as a proteoglycan. And these proteoglycans, the cell surface proteoglycans, are expressed by many different cell types. The proteoglycan function derives from the physiochemical characteristics of the GAG of the molecule, and that provides hydration and swelling pressure to the tissue, enabling to withstand compression forces. We see a lot of proteoglycans, and it's a major component of cartilage, so you can understand why, because it needs to actually withstand a lot of compression forces. For example, think the cartilage of our knees. Some example is the chorin, which is a widely distributed in many connective tissues proteoglycan, and has many different functions in regulating collagen fibril formation, and also in modifying the activity of transforming growth factor beta. Another proteoglycan, perlecan, the major heparan sulfate proteoglycan, is found in the basement membrane, and there it may play an important role as the major anionic site responsible for the charge selectivity in glomerular filtration. We'll be discussing a bit about this when we actually get to the basement membrane lecture. So, specific interactions between proteoglycans through both their GAG and the core protein component, and macromolecules in the ECM are the key factors in the functions of proteoglycans. So they exhibit biological functions, and they are just actually we get to know a lot more about them. We have more there, like more biological functions of proteoglycans are now gradually emerging. So, what are GAGs? Going back to GAG, to glycosaminoglycans, they're very useful in our bodies as a lubricant or a shock absorber. So it's an ideal lubricating fluid in our joints, and they also provide structural integrity to the cells. Um, they are um, they are quite important for during development and physiology. And I'm showing you here a schematic where it shows you how they are actually are able to act as lubricants or shock absorber. So I've already said that they're highly negatively charged. And that means that because they have this negative charge, they attract cations. Okay, an example would be sodium. And these cations are able to actually suck in water, and that creates pressure. So when that happens, when we have this actually absorbance of water, they enable ECM to withstand compressive forces. We're going to be discussing a bit about this and how that occurs later on with a schematic, just to show you what I mean. So this is how they are able to actually absorb shock and also act as a lubricant. So they are linear polymers. That means that they form quite large lines of disaccharides, or these repeating disaccharides. And they can be anything, the polysaccharide chain itself can be anything from 50 to 200 disaccharide repeats. So you can understand that they're quite long chains of sugars. So the structure of GAGs is specific, and it's specific to different family members. It depends on the exact type of sugar and the post-synthetic modification to the monosaccharide units. The type of modifications in GAG chains determine the proteoglycan function when the GAG is actually associated with a protein to make a proteoglycan. In addition, a major fact is that the sulfated GAGs have a high negative net charge. Here I'm showing you the disaccharides of four main GAGs, chondroitin sulfate, dermatan sulfate, and heparan sulfate and heparin. And if we look at what I just mentioned, the fact that the sulfated GAGs have a highly negative net charge, you can actually see here that if we compare a sulfated and an unsulfated GAG, such as hyaluronate, which is not sulfated, and heparin, you can see that the sulfate groups add the additional negative charge to the molecule.